Hi, my name is Shaumya and today we're going to be talking about polymers and monomers. This topic plays a major part in biology and can also be used in aspects of chemistry. It is important in biology because many aspects that we study in biology actually come from monomers and polymers. For example, when we look at genetics, we are actually looking at DNA, and DNA is a polymer. And in chemistry, when we go into organic chemistry and we start looking at alkanes, alkenes and alcohols, we're actually looking at polymers and monomers. Now let's move on and look at what a monomer actually is. So monomers are small individual units that are joined in order to form larger molecules, a bit like building blocks or Lego. So here we have a few examples of monomers. So first, let's start off with amino acids. So amino acids join together to form proteins, and proteins are vital for the body because proteins make up your organs, your skin, your muscles, and even your nails. Now nucleotides can also be found in the body because they join to form RNA and DNA. And RNA and DNA are very similar, but they do have some of their differences. For example, RNA is single-stranded, whereas DNA is double-stranded. Now let's look at these three. We've got glucose, galactose, and fructose. And these are a bit special because they're grouped together, and they can be referred to as monosaccharides. These are the monosaccharides. So glucose, when joined with another glucose, can form maltose. And galactose, when joined with glucose, forms lactose, and when fructose is joined with another glucose, it can form sucrose. Now let's move on and look at these monosaccharides in a bit more detail. So here we have it. At first glance, we can see that glucose and galactose are very similar, whereas fructose is different. Now what is this difference? So we can see that glucose and galactose look a bit like hexagons because of their six-sided shapes, whereas fructose more looks like a pentagon because of its five-sided shape. This is why glucose and galactose are called hexose and fructose is called a pentose. Now, glucose and galactose are very easily confused, and that's why in an exam you have to be extra careful. So when we look at this structure, we can easily mistake glucose for galactose, but we always have to look at this carbon here. Now, this carbon will tell us whether the glucose is actually a glucose or a galactose. Because here there's only one hydrogen, whereas here there's oxygen and a hydrogen. And in exam this is vital because say there's a question where it asks what glucose add glucose is, you have to make sure you're actually adding glucose to glucose. Because when you add glucose to glucose, we form maltose, but when we add galactose to glucose, we form lactose. So there's a big difference then. That's why you need to know the difference between the two structures. Now let's move on and look at polymers. So this is the name given to the molecules that are formed when many monomers are joined together. So here are the examples of polymers. So we've got the polysaccharides, which will be the many monosaccharides added together. Then we have proteins, which we discussed before and how important they are. Then we've got DNA and RNA. So what do you think the monomers would be for the polymers mentioned? So as we know before, for polysaccharides it will be monosaccharides, for proteins it will be amino acids, for DNA it will be nucleotides, and RNA will also be nucleotides. Now let's look at the reactions that are involved in polymers and monomers. So first we have hydrolysis. Hydrolysis is the reaction that occurs when a water molecule is used to break a chemical bond that has joined two monomers together. So as you can see in this diagram over here, this is maltose. So maltose is formed when two glucose is joined together. So we can see it is formed with this little bond here. Okay? So when you add H2O, which is commonly known as water, it then breaks back into two glucoses. Because as you can see, one of the oxygens and hydrogen attaches here, whereas the other hydrogen attaches here. And you can see they split into form two glucoses. So when water, so you can know this because it comes with the word hydro. And water has hydrogen and oxygen. So hydrolysis is a process in which water is needed in order to break something up. Now let's look at a condensation reaction. So condensation reactions is between two monosaccharides in order to form a glycosidic bond. And as a result of this, a water molecule is lost. So the difference between a condensation reaction and hydrolysis is that in a condensation reaction, water is lost, whereas in hydrolysis, water is gained. So let's look at the molecules here again. So again, we've got glucose. So we can see we've got the two OHs here. Now, when H2 is removed, so as we can see, H2 is added here and H2 is removed here. So when we remove the H2O, so let's remove the H2O. This little oxygen bonds both of these together. And this is called a glycosidic bond. And this is very important in monomers and polymers because this is how they're formed. So this is how a polymer is actually formed. So through this condensation reaction, polymers can be formed, and through this hydrolysis reaction, they can be broken. And the key element to both of them is water, because water is needed to break them, but water is given out when you make them. So normally when you need something to add more, but for this, you add more and it breaks it up. Now let's look at an exam question that you might get in an exam. So let's read this question. Cow's milk contains sugar lactose. Many cats are unable to digest cow's milk because they are lactose intolerant. Cow's milk can be made suitable for these cats by treating it with enzyme lactase to hydrolyze lactose. This makes the cow milk lactose free. Bees are coated with lactase and placed in a tube. As shown in the diagram below, cow's milk flows over the beads and lactose is hydrolyzed. So as we mentioned before, lactose is made up of glucose and galactose. So attaching lactase to the beads is more efficient use of lactase than adding the lactase differently to cow's milk. And now we need to look at 3 reasons why it's more efficient to attach lactase to the beads. So, for example, let's actually look at what the question's asking us. So they've given us a scenario where cats can't drink milk, and that's the basis of this whole question, that cats can't drink the milk, so we need to find a way for them to be able to drink the milk. So what's actually given us here is that enzymes are used, and they need to hydrolyze it in order for them to be able to drink it. So, why is it more efficient to actually use these beads? Now let's look at the mark scheme to see the actual reasons given. So here we are. So if we use beads, then these can be reused and not washed away. And you don't need to remove it from the milk. And it also allows a continuous process. So because you don't, as you're just using beads, you can just reuse them and start using them again. You don't have to filter it out, break the paper, then start something else. It's a continuous process. The enzyme is way more stable and it avoids end product inhibition. And what is meant by end product inhibition is that when we're left with the final product, it doesn't have anything unwanted inside. So even though this question is about enzymes and other stuff, it comes to know about lactose. And lactose is actually a polymer because it is formed when glucose and galactose join together. And if you didn't know what a lactose was and why this is all happening, then it will be hard for you to answer this question. And that's why polymers and monomers are very important in biology. Thank you.